Mitosis, a kind of cell division where daughter cells end up having the same amount of chromosomes as parent cells. Mitosis is important for the growth of human embryos. Don't forget the growth and development of our bodies. In mitosis, two identical daughter cells are being produced. This drawing explains the process of mitosis. Let's get to the first step. The first step, prophase. Prophase is by far the longest stage. In this stage, the DNA is condensing. The only thing holding the sister chromatids are the centromeres. If you notice, in this stage, the chromatids are getting paired up. As you can see, the membrane is starting to break down this stage. The black arrow is demonstrating how the spindle is starting to form, while the gray arrow shows the centromere holding the chromatids together. The second step, metaphase. In this stage, the spindles connect with the centromeres. The spindles then connect to their poles. The spindle fibers then line up the centromere to the equator. The red arrow is pointing at the centromeres, the black arrow is pointing at the spindle fibers, and the gray arrow is pointing at the centrioles. Step 3, Interphase. This is a stage where the chromosomes actually split. Every single split chromosome can now be called a daughter cell. They are split when the spindles thicken, and they pull when the spindles are being shortened. This causes the chromosomes to split. The fourth step, telophase. In this stage, the nuclear envelope starts to form again. The chromosomes then unfold back to a chromatin. Finally, the nucleoli starts to reappear. Now for the final step, cytokinesis. This sketch represents cytokinesis. In this stage, the chromosomes divide into two identical chromosomes. It splits after the nucleus reforms. I showed you what happens during mitosis. I never said what happens before it. There is a 3-step process that prepares for mitosis. It is called interphase. This wheel represents interphase. The G1 phase is the first phase of the 3-step process called interphase. In this step, the cells carry out activities to prepare for the next phase. Maturation of the cells also occur. It is a stage in which the cell cycle starts. The S phase is the second step in interphase. It comes after the G1 phase. In this step, DNA is checked and repaired if needed. The DNA is duplicated, which means it has double the amount of chromosomes, and then it moves to the G2 phase. The G2 phase is the final phase of interphase. There is an increase in the amount of RNA protein. The organelles that are needed for mitosis are produced. Finally, the cells are prepared for mitosis.